And that's it. I'm not going to put up with this anymore. You're in detention, buddy. Yeah. You're going to go over, cast yourself over to the Amazon Fire tablet. You did this, not me. Listen, I know it's slow as hell, but you'll think twice before you act like this again next time, won't you? Enjoy your 240p. Guys, you got to calm down. I can't teach with you just interacting like that. I'm gonna have to do a seating chart. I think this is the only way it's gonna work. All right, and there. I didn't want to do it, but you made me. All right, John, you're gonna look that way, and then Misha, you're gonna look that way, and then that way. All right. I've been wanting to do this for a while. All right, ready? Go. Here's the story of a lovely lady who was bringing up three very lovely girls, just like I imagined. Okay, for number 7, I think we're going to go ahead and multiply by 13. And Daniel, no, we don't talk sports right now. The Bucks would never get Brady. Are you serious? Are you serious? The Bucks, Tom Brady. If you're lying, I'm going to fail you. If you aren't lying, I'm failing everybody. Yeah, well, maybe if you weren't so lazy, things might be getting done around here. All right, guys, just click on the bullying prevention lesson. Let's go ahead and get started.
So what we're going to do here today is we're going to look at specific case studies, and these are going to be studies where a mutation of some sort in your DNA has resulted in a genetic disorder. And these are genetic disorders that are very widely talked about under the topic of genetics, right? There's quite a few genetic disorders out there that are very popular for teachers and scientists to talk about because they clearly demonstrate sort of the rule here of how this whole DNA-RNA relationship can influence your health. So to kind of introduce this concept or these case studies, I want to give an analogy here. So we're back in a factory, and so let's imagine that you are the foreman of a factory. All right. And so you are kind of on the ground helping the factory run, and it's a toy factory. It's the same as last time, right? It's a toy factory. And just the way that factories work is that you have some sort of central computer, and on that computer you have the information to make the toy. So it's kind of this digital blueprints, but they're blueprints that are encoded into the software on the computer. And so that information is used to direct the machinery, to direct the factory assembly line, and then they build the toys. Now, imagine that it's a Furby factory, if you guys remember that toy from the early 2000s. Some of you might not even have been born yet. But so if you remember Furbies, so let's say that you are you're at the factory and you're looking through the Furbies being produced, and one of them just has these weird like Barbie looking legs on it. It looks like one of those toys that Sid makes in Toy Story 1, that sort of thing. And you're just like, wow, that's really weird. Why did that toy come out weird? And let's say when you examine the rest of the toys, the rest of the toys are fine. It's just this one toy that was made by the assembly line that is odd. Now, what would you conclude? Would you conclude that something went wrong on the level of the blueprints in the computer, or would you just think that something in the machinery went wrong and produced this horrific toy? Right? Well, if you've thought about it a little bit, you'll realize that this is probably due to the machinery, right? Because most of the time, the machinery can be expected to get things right? The factory floor. It can be expected to get things right without messing things up, and most of them are correct. So this probably isn't an error in your computer or in your toy blueprints. But let's imagine a parallel situation. Let's imagine that instead you look at the toys, and most of the toys that come out are these messed up versions. And only occasionally does the normal toy get created by the factory line. All right, what would you conclude at this point? And I think if you're being logical here, I think you would conclude the opposite of last time. You would say, well, if what's going wrong here is happening most of the time, it's probably not the machinery's fault. It's probably because of some error in the information or in the blueprints. But notice, if there is an error in the blueprints, right, that error gets carried into the machinery, into the factory assembly line, right? The factory assembly line, it just does what the computer or what the blueprints tell it to, right? So if there's an error in the blueprints, that gets carried through the factory assembly line, and it ultimately results in an error in producing these toys. Now, this situation is what's really similar to genetic disorders, because genetic disorders are disorders that happen in your DNA, and your DNA is like the blueprints, the information, the software that makes you unique. But if there's an error here, that error gets carried all the way to your traits. So just to remind you, right, what we're talking about here is the DNA here in your body is kind of like the blueprints, right? It's your genetic information. Okay, it holds, it's a bunch of letters, right? Like it might be A, T, C, C, G, C, right? This is just information, but that information is used to build proteins. And remember, proteins are what give you your traits. They're what give you your phenotype. Now, here's the thing, is that proteins are the molecules that actually get stuff done in your body. They're like little molecular machines. They're like the toys, right? The toys are the things that actually get sold and actually get distributed to kids. And so here, proteins kind of serve that purpose. Proteins are the little machines that actually carry out the functions that are required in order for your body to stay alive. So if there's an error in your proteins, it might be because there has been some change in your DNA that was bad. So we're going to discuss two very common genetic disorders and discuss how changing the DNA actually results in the disease or disorder itself. So we're going to start with this, our first disease. We're going to discuss how blood works. Okay, so in your DNA, there is a gene called the hemoglobin gene, and there's actually 4 genes that code for hemoglobin. But specifically, we're talking about a gene called HBB. And so remember, a gene is just a stretch of DNA that codes for a protein. So this stretch of DNA right here is the hemoglobin gene because it is the information used to produce hemoglobin. And the hemoglobin protein looks like this. It's made up of 4 parts, and the way that it works is the hemoglobin protein is designed to absorb the oxygen that we breathe, O2. And so what happens is that these little heme groups, these little kind of teal groups that are in the middle, they have iron inside of them. And actually this protein, hemoglobin, that's what gives you your blood the red color that it has. Okay, so when you're looking at the red color of blood, what you're really looking at is this protein called hemoglobin. And hemoglobin carries oxygen, and it just is kind of a package carrier. It's kind of like a delivery man. It carries the oxygen to various parts of your body and delivers it to the cells who need it. So, like I said, hemoglobin is a protein inside of your blood cells that produces or carries O2 to your cells. All right, so this hemoglobin protein, like I said, is inside of your blood cells, but then your blood cells circulate throughout your body and carry the oxygen and things like that. Now, we're going to start talking about what's called sickle cell anemia. An anemia condition is a condition where something in your blood. So it might affect how much oxygen you get to your cells, how well you can breathe, stuff like that. And so let's take a look at the normal gene, the normal hemoglobin gene over here. So the normal gene goes, one side of the DNA is CCT, GAG, GAG. There's another side of the DNA here. Now that DNA gets transcribed into RNA, CCU, GAG, GAG, and then that codes for the amino acid proline, glutamic acid, and then another glutamic acid. Now understand something. The hemoglobin protein is much, much, much longer than 3 amino acids. Okay? It's actually thousands of amino acids long. All we're doing here is we're zoomed in on a part of the protein. So that DNA and those amino acids will combine together to form a hemoglobin protein. And that hemoglobin protein will then be inside of your red blood cell, and it holds the oxygen for your red blood cell. All right, but there's actually a mutation that happens in your DNA that causes sickle cell anemia. And here is the worst part. It is caused by one base change. Okay, the DNA gene that codes for hemoglobin is thousands upon thousands of bases long. And one change can cause sickle cell anemia. In this case, it's a substitution mutation because the A in the normal strand is switched out for a T. And so when it's transcribed, that error is carried over to the mRNA. It goes CCU, GUG, GAG. And instead of GAG coding for glutamic acid, GUG codes for valine. Now, that might not seem like a big deal, but what actually happens is that it changes the structure of the hemoglobin protein just a little bit so that it sticks to other hemoglobin proteins. So when those hemoglobin proteins stick together, they form these sort of long chains inside of your blood cell. And when they form those long chains inside of the blood cell, your blood cell begins to take on a sickle shape whenever it's not having oxygen in it. So when it does have oxygen in it, the blood cell looks normal. But whenever the blood cell gets rid of its oxygen, okay, this is no O2. When it gets rid of its oxygen, it turns into a sickle shape. And this is bad for your blood cell because your blood cell actually dies quicker. It dies more often. And these sickle-shaped blood cells tend to actually clump together in your blood vessel, and it creates a very, very painful condition for those who have it. So you can have sickle cell anemia. If you have sickle cell anemia, that means you have 2 bad copies of the hemoglobin gene. All right? Both of the copies that you got from your mom and your dad are both bad. But you can also have the sickle cell trait. Now, the sickle cell trait is someone who is heterozygous. So one version of the gene is okay, it's normal, but the other version is the bad hemoglobin gene. And there's actually an NFL wide receiver named John Brown. He played in 2017, he played for the Arizona Cardinals, but he had the sickle cell trait. So he was heterozygous for this condition. And it actually kind of affected him. He was having sore muscles and stuff like that. And he actually could only play later on in his career. He could only play every other game because of his sickle cell trait condition. So notice here that a single change in one of the DNA letters produces this trait called sickle cell anemia. So let's look at another example. A very common example, cystic fibrosis. So imagine that this is a lung cell. So this is the cell membrane inside one of your lung cells. So down here is inside one of your lung cells. Over here is outside one of your lung cells. And oftentimes in your respiratory tract, there's this sort of thick mucus that develops. And, you know, the kind of the phlegm you cough up. Now, this thick mucus is actually a good thing because it captures bacteria so that your body can get rid of it. But the thing is if it gets too thick, your body can't get rid of it very well. And so what happens here is that there is a protein that actually sits in the lung cell membrane, and the protein is called CFTR. Okay? CFTR. And so what CFTR does is it takes chlorine ions from inside the lung cell and it actually pumps them outside, right? So you pump the chlorine ions out here, and what happens is that those chlorine ions start mixing in with the mucus. Now, when this happens, this actually attracts water to the mucus. And so when that happens, the thick mucus actually starts thinning out and your body can clear it away. So that's what the CFTR protein does. It's kind of like a tunnel, and it's a tunnel that lets chlorine move through and thin out this mucus. Now, this is the normal working of CFTR. The condition cystic fibrosis happens whenever something bad happens to the CFTR gene. So I want to focus down here. So this is the CFTR gene. This is the normal gene right here. So the DNA goes ATC, ATC, TTT, GGT, GTT. And so once you transcribe that and translate it, the amino acids will be isoleucine, isoleucine, phenylalanine, glycine, valine. Okay? And notice, we're not looking at the whole protein here. We're actually at amino acid 506 and then 508. All right? Now, what happens during cystic fibrosis is that the gene for CFTR actually loses those three nucleotides, those three letters. They actually get deleted. Okay? So when you delete those three bases, okay, your sequence is now ATC, ATT, GGT, GGT. And so what actually happens is that an entire amino acid, phenylalanine, is gone. Notice it's the same, right? The amino acid sequence down here is the same. It's just missing phenylalanine. Phenylalanine should have gone right here, but now it got deleted. It's gone. And that might not seem like a big change, right? The CFTR gene is hundreds and thousands of amino acids long. Why would deleting one amino acid make a big difference? Well, let's look at the normal situation here, okay? In the normal CFTR gene, that protein, that CFTR protein gets created, and then what the cell does is the cell recognizes CFTR and then takes it and actually puts it in the cell membrane. Okay? But what happens when this mutation happens, what happens when this deletion mutation happens, is that a protein with a slightly different shape gets created. All right? But then what happens is that the cell, because of its slightly different shape, the cell does not recognize CFTR. And so because it doesn't recognize it, it actually destroys it before it can even get to the lung cell membrane. So it doesn't recognize this new protein, and it never puts it in the cell membrane. And so if there is no CFTR protein in the cell membrane, there is nothing to make this thick mucus thinner. And so what happens is that bacteria and all sorts of bad stuff get caught into this mucus, and your body can't get rid of it. And so that bacteria just sit in your lungs, and that's how you get lung infections. All right? So what's amazing about this entire process is how really tiny changes in your DNA can have huge consequences if you just follow the cause and effect. If you just follow the repercussions of that mutation, there can be huge consequences for even the smallest changes in your DNA. And so that's why mutations are really important for understanding medications, medicine, treatments, all that sort of stuff. So to summarize things a little bit, and this is probably the thing that Mrs. Cook and I want you to really understand through all of this is that changes or, mutations in your DNA carry over to the RNA and ultimately change how a protein is constructed. If proteins don't have the correct shape, they can't do their job, which causes diseases and disorders. Most things that could go wrong in the human body have to do with some sort of malfunctioning protein, and a lot of the times, those malfunctioning proteins can be blamed on some mutation that happens in your DNA.